Welcome everyone. So today in the lab we will be working on exploring the mutation rate for the specific gene CAN1. We will also be looking into various factors and conditions that may influence this mutation rate and why that may be the case. So to start us off with a trivia question, after reading through some background material on our experiment, what do you believe was the purpose of using the toxic arginine analog canavanine? If you chose answer choice B, you were correct. Canavanine, although toxic, shares many similar properties to the amino acid arginine, and this allows it to get transported from the surrounding medium into the cell through arginine transporters, which the CAN1 gene codes for. The toxic effects of canavanine causes the cell to stop undergoing mitosis and die. The colonies that are able to mutate the CAN1 gene will be resistant to the toxic effects of canavanine. To start off any lab work, it's always important to first clean up your lab station to prevent any confounding variables from affecting the experiment. To conduct this experiment, you will need a vortex. If you don't have access to one, I will show you alternative methods to homogenize your mixture later on. You will need a Sharpie, the wild-type culture that will be provided, pipette tips and pipettes, sterile water, sterile spreaders, 4 Eppendorf tubes, provided plates containing the YPD media and the SD media without arginine including canavanine, and sterile toothpicks. First start off with labeling your Eppendorf tubes and plates to ensure that you do not confuse them with each other and are able to monitor your progress and results. To measure the mutation rate for spontaneous mutation, each partner will only require one of each of these plates. Additionally, remember that later on, you will need a transilluminator and hydrogen peroxide solution for the later portions of this lab. Adjust the P1000 micropipette to 0.25. This will allow you to get 250 microliters of solution. Attach a fresh pipette tip and add 250 microliters of sterile water to your first tube or tube S0. Adjust the P1000 micropipette to 0.99. This will allow you to get 990 microliters of solution. Attach a fresh pipette tip and add 990 microliters of sterile water to tubes 2 and 4. Adjust the P1000 micropipette to 0.90. This will allow you to get 900 microliters of solution. Attach a fresh pipette tip and add 900 microliters of sterile water to tube number 3. Using a sterile toothpick, gently obtain a pin-size amount of wild-type yeast cells. You can see the amount demonstrated here. Submerge this into tube S0 or tube 1 containing 250 microliters of sterile water. Spread the culture inside the sterile water along the edge of the tube until most of the cells are off the toothpick. Vortex tube S0 or tube 1 to mix the cells fully in the sterile water. This tube should have a cloudy appearance. If you do not have a vortex available, that's no problem. Simply flick your tube until you begin to see a cloudy appearance and the cells are dissolved. You may use this flicking method in place for each vortexing step in the protocol. Keep in mind, you may have to flick many times in order to dissolve the cells fully. Adjust the P20 micropipette to 100. This will allow you to get 10 microliters of solution. Attach a fresh pipette tip and add 10 microliters of the content in tube S0 or tube 1 into tube 2, containing 990 microliters of sterile water. Make sure you vortex before and after each transfer. Keep in mind, you may also mix the solution with a micropipette by using the plunger on the micropipette to mix the two solutions together. Adjust the P200 micropipette to 100. This will allow you to get 100 microliters of solution. Attach a fresh pipette tip and add 100 microliters of the content in tube 2 and transfer it into tube 3, containing 900 microliters of sterile water. Make sure you vortex during each transfer. Once again, using your P20 micropipette, add 10 microliters of the solution in tube 3 into tube 4, containing 990 microliters of sterile water. Make sure you once again vortex during each transfer. Once your dilution is complete, obtain the YPD and SD minus arginine plus canavanine plates that you previously labeled. Adjust the P200 micropipette to 0.50 This will allow you to get 50 microliters of solution. Attach a fresh pipette tip and vortex tube S0 or tube 1 and add 50 microliters of the content in tube 1 onto the SD minus arginine plus cannabidine plate. Using a sterile spreader, slowly rotate the plate with one hand while moving the spreader up and down the plate lightly to evenly spread the material onto the plate. Next, vortex tube number 4 and once again adjust the P200 micropipette to 050. Attach a fresh pipette tip and add 50 microliters of the content in tube 4 onto the YPD plate. Using a sterile spreader, slowly rotate the plate with one hand while moving the spreader up and down the plate lightly to evenly spread the material onto the plate. Place all of these plates into the incubator at 30 degrees Celsius. Now that the spontaneous mutation experiment is complete, I would like you to begin thinking about how you believe adding hydrogen peroxide and UV will influence the mutation rate that you observe on your plates. For the hydrogen peroxide experiment, you must use 16 Eppendorf tubes. After properly labeling the tubes and adding 250 microliters of sterile water and a pin-sized amount of wild-type culture into each of the first 4 tubes, set aside the first tube, HP0. This tube HP0 is your control. You will not be adding any hydrogen peroxide into this tube. Next, adjust the P20 micropipette to 025, which will allow you to get 2.5 microliters of solution. Attach a fresh pipette tip and add 2.5 microliters of hydrogen peroxide into tube HP1. Vortex to mix the cells. Next, adjust that P20 micropipette to 100, which will allow you to get 10 microliters of solution. Attach a fresh pipette tip and add 10 microliters of hydrogen peroxide into tube HP2. Vortex to mix the cells. Adjust the P200 micropipette to 025. This will allow you to get 25 microliters of solution. Attach a fresh pipette tip and add 25 microliters of hydrogen peroxide into tube HP3. Vortex to mix the cells. Set your stopwatch to 15 minutes and press start. Leave these tubes at room temperature during the full 15 minutes to allow the hydrogen peroxide to take effect on the cells. You should now complete the rest of your dilution just as you had completed during the spontaneous mutation. Follow the same plating procedure for 4 YPD plates and 4 SD minus arginine plus cannabidine plates and incubate at 30 degrees Celsius. So now we'll be getting into the UV-induced mutation. Follow the exact same procedure demonstrated during the spontaneous mutation with 4 Eppendorf tubes. Follow the same dilution procedure along with the plating procedure onto 4 YPD plates and 4 SD minus arginine plus cannabidine plates. Once you obtain your transilluminator machine, recall that plates SD minus arginine plus cannabidine UV0 and YPD UV0 are your control. You will not be placing these plates under the UV lamp. Obtain the plates labeled SD minus arginine plus cannabidine UV1 and YPD UV1. Place them under the UV lamp for 5 seconds each with the lid off and the media side up. When you're done, replace the lid. It's important to note that you should only begin measuring time once the UV machine is turned on and the blue light is seen. It is recommended to use a timer for accurate time measurements. Next, you obtain the plates labeled SD minus arginine plus cannabidine UV2 and YPD UV2. Place them under the UV lamp for 10 seconds each with the lid off and the media side up. When you're done, replace the lid. Obtain the plates labeled SD minus arginine plus cannabidine UV3 and YPD UV3. Place them under the UV lamp for 15 seconds each with the lid off and the media side up. When you're done, replace the lid. Place all these plates into the incubator at 30 degrees Celsius. Once you finish conducting any of your lab work, it's always important to clean up your lab station for the next person to use. Well done, now it's your turn. If performed successfully, you'll be able to make observations about mutation rates, derive conclusions, and explore different factors contributing to mutation rates on the CAN1 gene. Follow the written protocol and remember to ask for help when needed.